Hello everyone! In this video, we will talk about the process of Krebs cycle. Krebs cycle is also known as tricarboxylic acid cycle. It is a biochemical pathway that is used to generate energy through the oxidation of acetyl-CoA. It is also used for synthesis of NADH and production of amino acids. Krebs cycle takes place in the mitochondria of the eukaryotes and in the cytosol for the prokaryotes. The first step involved is a preparatory step which starts with the pyruvate. Pyruvate is derived through the glycolysis of glucose, which is a 6-carbon compound, and is split into two molecules of pyruvate, which is a 3-carbon compound. The next step includes the oxidation of pyruvate into acetyl-CoA by the enzyme pyruvate dehydrogenase complex. And in this reaction, a molecule of carbon dioxide and a molecule of NADH is generated. The acetyl-CoA is a 2-carbon compound. In the next step, the acetyl-CoA is combined with oxaloacetate, which is a 4-carbon compound, to form citrate, and hence the resulting molecule is a 6-carbon compound. This reaction is catalyzed by the enzyme citrate synthase. In the next step, the citrate is isomerized into isocitrate by the enzyme aconitase. Next, the isocitrate is oxidized into alpha-ketoglutarate by the enzyme isocitrate dehydrogenase. And in this reaction, a molecule of NAD is reduced into NADH, and a molecule of carbon dioxide is generated. Hence, the alpha-ketoglutarate is a 5-carbon compound, since one molecule of carbon is lost into carbon dioxide. Next, the alpha-ketoglutarate is converted into succinyl-CoA through the enzyme alpha-ketoglutarate dehydrogenase, and in this reaction, a molecule of NAD is reduced to NADH, and a molecule of carbon dioxide is released, hence making succinyl-CoA a 4-carbon compound. In the next step, this succinyl-CoA is converted into succinate by the enzyme succinyl-CoA synthase. In this reaction, a molecule of GTP is generated. Next, succinate is converted into fumarate by the enzyme succinate dehydrogenase. In this reaction, a molecule of QH2 is generated, which is used for the production of FADH2. Fumarate is then converted to malate by the enzyme fumarase, and in the last step, the malate is converted into oxaloacetate by the enzyme malate dehydrogenase, and in this reaction, NAD is also reduced to NADH. Now, let's look at the result of the Krebs cycle. Through each cycle of the Krebs cycle, the following products are generated: 3 molecules of NADH, 1 molecule of FADH2, 1 molecule of GTP, and 2 molecules of carbon dioxide. Now, since glucose is split into 2 pyruvate compounds, for each molecule of glucose, this cycle runs twice. Hence, the products formed are 6 NADH, 2 FADH2, 2 GTP, and 4 carbon dioxide. All the NADH and the FADH are next fed to the electron transport chain for the generation of ATP. That is all for the Krebs cycle. Thank you for watching.